Whorls, what is it called? Epigynous. If the ovary is below the rest of the whorls, it is called hypogynous. Epigynous, perigynous, hypogynous. It is the position of the ovary. The specific palindromic sequence which is recognized by ECO R1 is GAA, TTC, GAA, TTC from both sides. That is the recognition site for ECO R1. You have to remember this recognition site. Different restriction enzymes will have different palindromes that it identifies. ECO R1, you have to remember. This is a thing that might come as a question that you have to remember. GAA, TTC. Wrong statement with regards to restriction enzymes. Okay. Each restriction enzyme functions by inspecting the length of a DNA sequence. True or false? Does the restriction enzyme inspect the length of the DNA sequence to identify where it has to act? Yes. They cut the strand of DNA at palindromic sites. Is that correct? No? Then where do they cut?
A little away from the
restriction enzyme?
A little away from the palindromic site.
Oh, we're going technical. Palindromic site is what it identifies, no? It's okay, let us keep it that way. Let us see. To keep you happy. They are useful in genetic engineering. Correct? Sticky ends can be joined by using DNA ligases. Statement is correct. But is it anyway related to restriction enzyme activity or restriction enzyme? It is a property of ligase. It is a property of ligase. Sticky ends come together. In fact, it is a property of the base pairing. The complementary base pairs will form hydrogen bonds. That's the property. So, between our friends' technical point of they cut the strand of DNA at palindromic site being wrong because it cut it outside of the palindromic site versus sticky ends can be joined by using DNA ligases. Which one do you want to choose as the wrong statement with regard to restriction enzymes? Option 4. We can choose option 4. Though it is something that happens after the restriction enzyme has done its job. The question is related to what is not right about restriction enzyme. The statement is not related to restriction enzyme only. Which of the following is put into an anaerobic sludge digester for further sewage treatment? Okay, which of the following is put into what is activated sludge? What is activated sludge? See, there are different statements given here. Primary sludge, floating debris, effluents of primary treatment, activated sludge. In sewage treatment, what are all this? What is primary sludge? What is sludge in the first place? What is sludge? All the suspended particles that you find in sewage is sludge. Floating debris is that which floats. Whatever debris that is floating. Effluent is what? Effluent is waste or treated water. You have treated water which is purified to a certain extent. Effluent is the treated water or the waste. Effluents are thrown or effluents are reused. Effluents are reused. Effluents are treated further. After removing all the debris, you have the effluent treated further for purification. Okay, what is activated sludge? Sewage, after you remove all the debris, you aerate it. You put a lot of air into that sewage. You churn the treated sewage with air, oxygenated. Then whatever settles down at the bottom is activated sludge. It is a microbial rich mass. It's a rich mass of microbes. So anaerobic sludge digester is, you put activated sludge into the anaerobic sludge digester for further treatment. Alright. Correct events that occur during inspiration. Select the correct events. Okay, contraction of diaphragm. Does it happen? Yes. Contraction of external intercostal muscles. Yes. Pulmonary volume decreases. What happens during inspiration? Pulmonary volume increases. Intrapulmonary pressure increases. Intrapulmonary pressure decreases. So A and B are correct events that happen during inspiration. C and D are the events that happen during expiration or exhalation. You should, the correct way of terminology is inhalation and exhalation. Inspiration and expiration are not very appropriate. Okay. What happens to the shape of the diaphragm during inhalation? Diaphragm becomes flattened. Diaphragm becomes flattened. Diaphragm contracts or relaxes for inhalation to happen. Is contraction or relaxation giving rise to inhalation of diaphragm? Contraction. So diaphragm flattens. What happens to intrathoracic pressure during inhalation? Intrathoracic pressure, not the intrapulmonary pressure, intrathoracic pressure during inhalation. Huh? Increases, huh? Pressure. The intrathoracic pressure decreases, the intrathoracic volume increases. See, the intrapulmonary volume or pressure is a reflection of the intrathoracic volume or pressure. It's directly being transmitted. What is the kind of movement that you see of the ribs during inhalation? They move out and up. So how do we refer to that? Bucket handle movement. What is the movement of the upper ribs during inhalation? They move up and down. What kind of movement is this? Pump handle movement. Two types of movements are references to the movement of lower ribs and the upper ribs during inhalation. Bucket handle movement and pump handle movement during inhalation and exhalation. If the head of a cockroach is removed, it may live for a few days because cockroach anyways does not think. Right? The nervous system of cockroach is not entirely dependent on the brain, which is a very small part. It is distributed throughout the body on the lateral part in the form of ganglia. And those ganglia will help it continue to survive and maintain all other functions. But why does the cockroach die then? No food. What is the right choice? 3. The head holds a small portion of the nervous system while the rest is situated along the ventral part of its body. See, even for all other organisms also, not only cockroach or anything, we have intelligence at every part of our body. Every part of the body is intelligent. Nervous system is kind of a processing center, but every part of the body is intelligent. The liver decides how it should function at its own level. There is gut intelligence also. There is connection of gut to the brain. Have you heard of something called gut-brain axis? Gut-brain axis. There are a lot of microorganisms in the gut. The microbial flora within your gut determines the kind of stimulation that happens in your brain. So, that connection, so it's the right kind of food that you have to constantly eat. Prebiotic foods, probiotic foods, lot of fermented foods. Does not include alcohol, okay? Though it is fermented drink. Okay. A lot of fermented foods will help in nurturing the gut bacteria, which produces many chemicals which act on the brain also. So your mental health depends on your gut health. Your physical health depends on your gut health. Big connection between your gut, what you eat and how you are. So there's a saying, no? Tell me who your friends are, I'll tell you who you are. It can be made more evolutionary. Tell me what you eat, I'll tell you what you be, what you are. That's all. And eating should not be, eating is one, food. Your entire life is determined by what you consume. Eating is a part of your total consumption. Consumption through all sense organs also should be paid attention to. Intellectual development is dependent much more on the sensory consumption than dietary consumption. You can eat anything that you want, as long as your physical health is being maintained, that's fine. But your sensory consumption determines your intellectual development. Your growth is determined by dietary consumption. Development is supported by dietary consumption, but intellectual development is influenced by sensory consumption. What you consume through your sense organs. Very important. Which of the following statements are true for the phylum Chordata? In Eurochordata, notochord extends from head to tail and is present throughout their life. Now here, you have to look at, is something a part of chordata? Then there are options talking about notochord. So is notochord present? If it is present, for how long it is present? All of that we have to look at. Statement A, is Eurochordata a part of chordata? There are three classes in chordata. What are they? Eurochordata, cephalochordata and euchordata or chordata or vertebrata. Eurochordata is chordata, but it has notochord, which is a characteristic feature of chordates. Does it extend from head to tail and present throughout life? Eurochordata. It is present only during the larval stage. In vertebrata, notochord is present during the embryonic period only. That is correct. That is correct. Okay, I will make another statement. Spinal cord is modified notochord. Spinal cord is derived from notochord. True or false? False. Spinal cord is derived from ectoderm, the neural ridge. But spinal cord development is guided by notochord. Notochord gives the axis and the direction. Notochord gives the axis and the direction for the development of the nervous system. Notochord itself is not a part of the nervous system. Alright? So cord, you should not consider it to be same. So statement B is correct statement. Look at the options which includes B. 1 and 2 does not include B, rule out. A is wrong, so anything with A should be ruled out. Anything without B should be ruled out, anything with A should be ruled out. So we got our answer. Option 4. Check with C. Central nervous system is dorsal and hollow. That is correct. Chordata is divided into 3 subphyla: hemichordata, tunicata and cephalochordata. Hemichordata is not chordates, it's a separate phylum. Tunicata is, are tunicates chordates? Tunicates are chordates, but hemichordata is not chordate. Cephalochordata is also chordata. Okay. Organism with its use in biotechnology. Bacillus thuringiensis, triprotein, Thermus aquaticus, Construction, thermos aquaticus, DNA polymerase, there is Taq polymerase, Agarobacterium tumefaciens, cloning vector, Salmonella typhimurium, construction of first rDNA molecule. rDNA is the common DNA. So, option 2. (28), 29. 28 is out of syllabus now. I'm very happy about the mineral nutrition chapter being out. I had also figured out how to remember the macronutrients, micronutrients and all. I think I have told you earlier. Have I told you? Macronutrients are CHON PK. CHON, CHON, PK. Boom right. He's roaming around after having a drink. CHON PK. Incorrect statement. Heartwood does not conduct water but gives mechanical support. Is it right? Heartwood. Oh. Match the following. Heartwood has been rejected now. But you can't break it. It's very bad. Matching. Inhibitor of catalytic activity, possess peptide bond, cell wall material in fungi, secondary metabolite. Which one do you want to catch hold of? Cell wall material in fungi. So which is? Chitin. Chitin is 3. C should be 3. Alright. Then A should be inhibitor of catalytic activity, malonate, possess peptide bonds, collagen. Collagen is the, what is special about collagen? Just if you look at collagen, what all comes to your mind? Collagen. Anything? Huh?
Connective tissue.
Collagen is a major component of connective tissue. Okay then. All connective tissue has collagen as the matrix, in the matrix. Then collagen is the most abundant protein in the animal kingdom. Collagen is the most abundant protein in the animal kingdom. Okay. Next question. Ricin is the secondary metabolite, it's a toxic protein. Meiotic division of the secondary oocyte is completed. We did this now again in the same paper? No, the previous paper. But today only we did this. Same question. Meiotic division of the secondary oocyte is, in the previous paper it was asked, when is the second polar body separated? Now it is asked, meiotic division in the secondary oocyte is completed when? At the time of fusion of a sperm with an ovum. What gets completed? Meiosis 2. According to Robert May, the global species diversity is about 7 million. How many have been described? 1.5 million. Okay, whatever we have described we know because there is a number for it. It has been described already. 1.5 million, there cannot be any ambiguity. 7 million, how do you say that? There will be some scientific way of arriving at that conclusion. It is not like telling how many grains of sand are there in this, my fistful of sand. You tell some number. I say no. Then what will you say? Count. All lame jokes. I enjoy a lot of these small jokes. But I am criticized for telling so many senseless or poor jokes. My sister has given a brand of jokes. What do you call a bad joke? You call it a PJ, poor joke. Alright. My brand of jokes are apparently bad than that. It is referred to as VJ. Varun's jokes. It's okay, I don't bother about what people think as long as something is making me happy. The first phase of translation is binding of mRNA to ribosome, recognition of DNA molecule, amino acylation of tRNA, recognition of an anticodon. What is the first phase of translation? Amino acylation. Now, amino acylation begins when after the binding of the mRNA, after the binding of the smaller unit of the ribosome to the mRNA. Smaller unit of the ribosome binds to the mRNA. That activates the tRNA which is supposed to get methionine. Then that tRNA catches hold of amino acid. That tRNA catches hold of amino acid, methionine, and then there is amino acylation. Why shouldn't we consider binding of mRNA to the ribosome as the first phase of translation? Why shouldn't we consider? The answer given is amino acylation of tRNA only, no? In the answer key also. That is what we should consider also. But why should we not consider the first option? Only when the entire machinery is assembled, you begin the process. You don't consider the assembly, the process when the assembly is happening as a part of translation. Translation is when you actually start translating the codon into amino acids. So what is the first step there? You have to catch hold of the amino acid by the tRNA has to catch hold of the amino acid. So amino acylation of tRNA is technically the first step. Or like process wise, it is the first step when it comes to translation. Okay. We'll do till 35 and then take to break. Which of the following regions of the globe exhibit? Okay, you tell me the three sites on the ribosome for translation. Amino acyl site, then where amino acid carrying tRNA comes and binds, that is the A site. The next site is P site, or it's also called the peptidyl transferase site. Peptidyl transferase is active in the P site. Then the next site is E site or the exit site. Okay. There is a ribozyme associated with the translation. Name the ribozyme. Name the ribozyme. Huh? Name the ribozyme, not the chemical nature of the ribozyme. Peptidyl transferase. Peptidyl transferase is the enzyme. Ribozyme is a reference to some enzyme. You should tell the enzyme. The question is, what is the chemical nature of that? It is ribosomal RNA. What is its size? Then it is 23S ribosomal RNA. Name the stop codons. Opal, ochre, amber. Name the codon. Give me the genetic code of the codons. UAA, UAG, UGA. UAA, UAG, UGA. In the same order, opal, ochre, amber. Which of the following regions of the globe exhibits the highest species diversity? Western Ghats of India, Madagascar, Himalayas, Amazon forests. Are all the four biodiversity hotspots? Western Ghats, Madagascar, Himalayas, Amazon forest are all biodiversity hotspots? No, which one is not? Huh? Madagascar is not. Why do you think the movie is made with the name Madagascar? It's biodiversity hotspot only, not in India. Which are the biodiversity hotspots in India? Indian subcontinent. The Himalayas, Western Ghats, then Indo-Burma region, then anything else? Sri Lanka also. Sri Lanka is also considered to be a part of the Indian subcontinent. Like European Union, we should have a Indian subcontinent union. Difficult. Which of the following statements is not correct? In man, insulin is synthesized as a proinsulin. Correct or wrong? It is correct. There are three peptide bonds, A, B, C. The C peptide bond is the link between peptide A and peptide B. You have to remove the C peptide to make it active insulin. The proinsulin has an extra peptide called C peptide. That is also correct. The functional insulin has A and B chains linked together by hydrogen bonds. That is wrong. What is the bonding between the two chains of insulin? Disulfide bonds. It is disulfide bonds, not the peptide bond. Genetically engineered insulin is produced in E. coli. That is correct. Okay, which is the company that produced it first? Eli Lilly. Eli Lilly. Okay, take a break. 5 minutes, not for sunbathing, just for washroom break. Sleep break, not sleep break. To overcome sleep. Plant being monocot plant, monocot stem. So let us look at the others. Large conspicuous parenchymatous ground tissue, that is also characteristic of monocots. Vascular bundles conjoined and closed. Conjoined and closed vascular bundles, monocot again. In dicots it is open vascular bundles, because open vascular bundles will allow for secondary growth. Phloem parenchyma absent, that is also monocot. So identify the category of plant and its part. Monocot stem. Matching again. Which are you sure of the four? Poison sting, trigon. Trigon is stingray. Trigon is stingray. So D should be 1. You got 50-50 by this now. What else you can look at? 6 to 15 pairs of gill slits. Cyclostomes. So A should be 2. A to D1 is option 1. Air bladder also is easy. Bony fishes have air bladder. Cartilaginous fishes do not have air bladder. Air bladder. From his experiments, Miller produced amino acids by mixing the following in a closed flask. Look at the options closely. Methane is there, ammonia is there. So 2 options give methane, 2 options give CH3 and CH4. What did he mix? CH4. He used methane, CH4, methane, hydrogen and ammonia. Methane, hydrogen and ammonia and water vapor. 2 options have the same, except temperature being 800 degrees one option, option 1, and 600 degrees in third option. Which is more reflective of the early Earth's atmosphere? 800. 800. There is one more which he introduced into this flask. Which is that? Apart from these things, methane, hydrogen and ammonia with water vapor at 800 degrees, something more heated. Electrical spark to mimic lightning. Embryological support for evolution was disapproved by. Who disapproved embryological support for evolution? One pair. What is the embryological support that for evolution? Haeckel's recapitulation theory. recapitulation theory. It is also referred to as biogenetic law. What follows what? Phylogeny follows ontogeny, or ontogeny follows phylogeny? Ontogeny follows phylogeny, or ontogeny recapitulates phylogeny. Ontology refers to individual. Phylogeny refers to evolutionary history. Ontogeny refers to the individual history, organism's history or life cycle. Phylogeny refers to the evolutionary history. Process responsible for facilitating loss of water in liquid form from the tip of grass blades at night and in early morning is. Previous paper we discussed this. What is the process? Root pressure. Imbibition is seen in what? What displays imbibition? Generally, seeds. Seeds, germination of seeds is facilitated by imbibing water. Imbibing water. Do you remember the pore at the tip of the leaves through which dewdrop is formed? What is the pore called? Hydathode. What is the process called? Guttation. Secondary metabolites such as nicotine, strychnine and caffeine are produced by plants for nutritive value, growth response, defense action, effect on reproduction. They are all produced as defense. They are all produced as defense. The oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of one molecule of a 4 carbon compound and one molecule of a 2 carbon compound. Rubisco is primarily responsible for what? What is it participating in? In fixing carbon dioxide into a 3 carbon phosphoglycerate, which will again combine to form. What is the first hexose produced? Is it glucose that is produced first? What is produced first? Fructose is produced, which will again get isomerized into glucose. So it is not a 6 carbon glucose that is produced as a byproduct, as the first stable product of photosynthesis. It is a 3 carbon compound, 3 phosphoglycerate that is produced, which will again combine to form 6 carbon hexose, glucose or fructose and then glucose. What is the energy balance sheet for Calvin cycle? How many ATPs and how many NADPH are used? 18 ATP and 12 NADPH is used for producing one molecule of glucose. BT cotton variety was developed by the introduction of toxin gene of Bacillus thuringiensis and is resistant to insect pests. Bollworm. What is the toxin? Critoxin. Why is it called critoxin? The toxin is a crystalline protein. Because the toxin is a crystalline protein, it is referred to as critoxin. Which of the following refers to correct example of organisms that evolved due to changes in environment brought by anthropogenic action? What do you mean by anthropogenic action? Human interference. Anthropology is the study of humans. Anthropology is the study of human and humans' interaction with the surroundings. So anthropogenic action means action generating out of human activity. Now you look at the options. Darwin's finches of Galapagos Islands. Is it natural or artwork? Natural. So not anthropogenic. So So it's brought about by changes in the environment and not by human action. The other three are human interference. Herbicide resistant weeds. Why are they developing? Because we are using herbicides, giving opportunity for the weeds to develop herbicide resistance. Drug resistant eukaryotes. Drug resistant eukaryotes, again anthropogenic action. Man created breeds of domesticated animals like dogs. They are also anthropogenic action. So B, C, D are the ones which are brought about by anthropogenic action. Wrong statement with reference to immunity. When exposed to an antigen, antibodies are produced in the host's body. This is called active immunity. Is it right or wrong statement? Correct. That is active immunity. You need exposure to antigen. Body will develop the ability to produce antibody. Active immunity. Okay. See again, it's like correlated with content-centric learning and competence-centric learning. You have to master the content to do well in exams. If you look at examination, doing well in examination as being equivalent to fighting the disease. Fighting the disease, if you compare both. What the teacher gives you is passive immunity. Helpful only during emergency. You should use it only during emergency. What is to be done throughout your attempt with learning? You should try to develop active immunity. What is that? You get exposed, develop the ability to deal with it, and then you deal with it, and then you will develop the ability to deal with it. Correct? What you did from the beginning? Starting from the syllabus map, level 1 notes, multi-perspective notes, post-test analysis notes, all of that. Now I am telling you, I will give you the notes that you will read for 1 month and do well in exams, get 300 plus marks. Passive immunization is helpful only to tackle the immediate demand. If your entire dependency is on passive immunization, someday some challenge will kill you. You will not be able to deal with all challenges. So developing the ability to face challenges is evolutionary. That is natural. Using tools to face challenges is manipulation. It will help you win the challenge, but it will not make you capable of winning every challenge that you face. So if you can win the challenge also, while you develop the ability to face challenges, which one do you want to attempt? Do you want to make the short-term challenge easier by using tools, or put in a little bit more effort to develop the ability to make tools so that the tools can be used for every challenge in life? See, active immunity is important. Passive immunity only as a survival strategy, only as a survival intervention. But who wants it? Everybody wants less effort, more outcome. And the tragedy of life is, see again, as a doctor, you should treat patients by a prudent approach. You should not just give antibiotics left, right and center. You should not give antibiotics without the absolute need for antibiotics. And you should not use steroids unless there is a pressing need for it. But antibiotics will make you get down the load of infection quickly. If you treat the symptoms and allow the body to fight the disease for a while, you will recover still, but you are managing symptoms with medicines that you give the patient. But it will take a few days more for the patient to recover. In this approach, you will allow the patient to improve his immune status also by supporting the immune system to fight the disease. You are giving antibiotics and fighting the disease on behalf of the immune system. But the patient recovers, starts feeling better on the first day, or the first or second day when he gets the antibiotic. You give steroids, suddenly inflammation goes down, and suddenly the patient starts feeling good. So who will be considered a good doctor? The one who makes you feel better quickly, or the one who boosts your immune system by the right approach. Market forces. Alright, as a doctor and as a teacher, you should never be driven by market forces. You should be driven by what is right. Do what is right. Develop your ability to convince people why it is right. Or you should develop the communication ability to convince people. Tell people, look, this is right because this is how it works. At the end of the day, people want the good things to happen to them, no? Would your patient want to recover from this illness along with developing a better ability to not fall sick subsequently? Or does your patient tell you, I don't care about falling sick later, but right now I want to just get well today only? What do they choose? Do they choose 1 or 2 days more of a little bit discomfort for a better strength to fight diseases in the future? Or to cut short this, will they want to get exposed to more diseases and again come back to the same situation later? Likewise, as a teacher, if you are training the students to become capable of self-dependence, and that also gives them ability to tackle the exam which is of immediate priority, wouldn't people understand? So stick to doing what is right, not what everybody expects you to do or what everybody is doing. Convenience is easy to choose. But if you know what is right and you choose convenience, you are not a right person. You should have the strength to fight for what is right. Okay. When ready-made antibodies are given, it is called passive immunity. Is it correct? Yes. Active immunity is quick and gives a full response. Is it right? No. Active immunity is slow and gives a graded response. It does not give a full response. Repeated exposure is required. Like revision. Revision is booster dose. The fetus receives antibodies from the mother, which is an example of passive immunity. That is correct. Statement 3 is wrong. By which method was the new breed Hisardale of sheep formed using Bicanary ewes and Merino rams? Crossbreeding. Both are sheep only. So of 2 different breeds, you breed 2 breeds and come up with a new breed. It is crossbreeding. What is outcrossing? What is outcrossing? Same breed but unrelated lines. Same breed is but unrelated heredity. At least for 3 generations they don't have common ancestors, but the same breed. It is outcross. Outcross and crossbreeding. Outcrossing is within the same breed. Inbreeding is related individuals. They have common ancestors. That is inbreeding within the same breed. Correct statement with reference to human digestive system. Next question. Out of syllabus, no? Match the following. What do you want to catch hold of? This is repeating again and again, same things here and there. What do you want to catch hold of first? Clostridium butyricum. Okay. A2. Butyric acid. Only option is option 2. Check for the rest. B1. Trichoderma polysporum cyclosporin. Cyclosporin is used for immunosuppressant. Immunosuppression is used as an immunosuppressant. Monascus purpureus. What does it produce? Statin. Aspergillus niger. Citric acid. Done. Presence of which of the following conditions in urine indicates diabetes mellitus? Uremia and ketonuria, uremia and renal calculi, ketonuria and glycosuria, renal calculi and hyperglycemia. Ketonuria and glycosuria. Okay, uremia is what? Increased urea in blood. Emia is a reference to blood. Alright. Increased uric acid in blood. That is uremia, uric acid in blood. It is not an indication of diabetes, it might be an indication of renal failure also, or it might be an indication of some protein metabolism defect in the liver. Alright? Ketonuria is what? Excretion of ketone bodies in the urine. What does that indicate? How is it a reflection of diabetes mellitus? What does ketonuria indicate in the first place? It tells you that lipids are being used for metabolism. Lipids are being used for metabolism. Have you heard of keto diet? Keto diet, where you don't eat any carbohydrates, you eat a lot of fat, and the body goes into fat metabolizing mode, breaking down fat, and ketone bodies are excreted. Ketone bodies are a byproduct of fat metabolism. So ketonuria is telling you, it is telling you just that there is ketone bodies in urine. That means fat is being metabolized for energy. If fat is being metabolized for energy, what does that mean? Sugar is not available. Glucose is not available for metabolism. Likely cause, non-pathological cause can be, the person might be in starvation. The person might be in starvation. It is your patient. You can ask him. If he is in front of you, you can ask him, have you not eaten food? If the person is in starvation, ketonuria might be an indication of starvation. But ketonuria with glycosuria. Glycosuria is what? Presence of glucose in urine. Why is glucose coming into urine? Because there is excess glucose in blood. There is much more glucose in blood, which after being filtered out into the nephron, cannot be completely taken back. So there is excess glucose in blood which is getting excreted in the urine. There is more glucose in blood, but the patient's body is using lipids for energy needs. It is breaking down lipids indicated by ketone bodies. What does that mean? The body is not able to use glucose. Why is it not able to use glucose? The body is not having insulin to tell the cells to use glucose. So that is indicative of diabetes. Why ketonuria and glycosuria together is very significant. Why ketonuria and glycosuria together is significant is because of that. Alright? What else? Renal calculi has got no significance, has got no relevance to diabetes as such. Alright? Next. Floridian starch has a structure similar to starch and cellulose, amylopectin and glycogen, mannitol and algin, laminarin and cellulose. First of all, Floridian starch is produced by what? Floridian starch is produced by red algae. It is the storage carbohydrate of red algae. And Floridian starch is a branched polysaccharide. Which of these is branched? Amylopectin and glycogen are branched polysaccharides. So Floridian starch is similar to amylopectin and glycogen. Select the option including all sexually transmitted diseases. So the option should have only sexually transmitted diseases in it. Check for the ones which is not a sexually transmitted disease and you will rule out those options. Which ones are not sexually transmitted among the given options? Malaria and filariasis. Malaria, filaria, cancer, all three are not sexually transmitted. So, gonorrhea, syphilis and genital herpes are sexually transmitted diseases. Just a moment, somebody is repeatedly calling in spite of receiving the call. Repeated calls, if I am expecting a call, I'll receive. Or if it is from family, I'll receive. Unknown calls, we'll see. Matching again. See again, cell biology question. It has to come with cell cycles only. Either mitosis or meiosis. Sometimes direct question, sometimes we match the following. Which one do you want to catch hold of? Huh? Zygotine. Okay, what happens in zygotine? Synapses. So A4. Only one option. Second option. Zygotine, synapses. Pachytine, crossing over. Diplotine, chiasma formation, diakinesis, terminalization. One phase is left out in this. Leptotine. What happens in leptotine? I want you to give me the technical word for what happens in leptotine. What is formed in leptotine? Bivalent. Leptotine, bivalent. Pachytine or zygotine, synapse. Pachytine, crossing over. Diplotine, chiasma. Diakinesis, separation or terminalization. Which of the following pairs is of unicellular algae? Laminaria and sargassum, Gelidium and Gracilaria, Anabina and Volvox, Chlorella and Spirulina. Chlorella and Spirulina are unicellular algae. Laminaria and Sargassum are brown algae, Phaeophyceae. They are not unicellular but they are multicellular. Gelidium and Gracilaria are red algae, Rhodophyceae, again multicellular. Gelidium and Gracilaria are used for production of agar. Gelidium and Gracilaria are used for the production of agar. Anabina and Volvox. Anabina is cyanobacteria, filamentous, Volvox, green algae, colonial green algae. They are not multicellular, but they remain in colonies. But Anabina is filamentous, so which hormone levels cause ovulation from the Graafian follicle? High concentration of estrogen, high concentration of progesterone, low concentration of LH, low concentration of FSH. Now, what is responsible for ovulation? The peak of which hormone is responsible for ovulation? The peak of LH is responsible for ovulation. But peak of high concentration of LH is not given in these four options. What do you want to choose now? Estrogen. Because estrogen continues to rise until ovulation. Estrogen continues to rise until ovulation. So you can consider that as well. But estrogen is rising until ovulation, but estrogen is not the one responsible for ovulation. Ovulation is brought about by increased levels of LH. But estrogen triggers a spike in LH, which will lead to ovulation. Again, matching. Lot of matching questions in this paper. Bt cotton, adenosine deaminase deficiency, RNAi means what? RNA interference, PCR. Which one do you want to catch hold of? PCR. Okay, you want to catch hold of Bt cotton? Bacillus thuringiensis A4. Correct? Look at the options. They have given only one option with A4. Some kind person setting the paper. A4, adenosine deaminase, gene therapy, RNA interference for cellular defense, PCR, detection of HIV infection. Montreal Protocol was signed in 1987 for the control of out-of-syllabus questions. Montreal Protocol, again, we have done this 3 times in the last 2 days. CFC, chlorofluorocarbons. Which of the following is correct about viroids? Free RNA without a protein coat. Viruses, viroids, prions. In the same order. Viruses, genetic material with protein coat. Only genetic material. viroids. Can it be DNA also? Can viroids have DNA also? No, free DNA is not considered viroids. It is free RNA only. Then you have prions, which is proteins, abnormally folded proteins. The ovary is half inferior in plum. You have to remember, that's all. Brinjal and mustard apparently superior ovary, sunflower is inferior ovary. Always. The enzyme enterokinase helps in the conversion of, again, enterokinase is digestion. Digestion is out of syllabus. Look at the happiness. What if questions come? You will raise very strong objection and make them give you grace marks. So if it is out of syllabus question, do you want to attempt it or you don't want to attempt it? Attempt it. But still tell me, you have studied this no earlier until last year you were studying this? No? Okay. Match the trophic levels with species examples in the grassland. Trophic levels 1, 2, 3, 4. The four animals given are at 14 patches of land. 14 patches in which he was cultivating pea plants in a controlled manner. Match the following columns. Match the following. In water hyacinth and water lily, pollination takes place by water hyacinth and water lily. Insect pollination and water also, no? No water? Only insecta? Water hyacinth and water lily? Entomophily. Is hydrophily not possible? Water hyacinth and water lily are both hydrophytes, no? They are water plants. Is pollination by water not possible? Why? The flowers are outside of the water surface. Hydrophily is not possible, it is entomophily. Name the plant growth regulator that increases the length of sugarcane stems, thus increasing yield. Gibberellin. What is the disease caused due to increased gibberellin production by a fungus? Bicanera. Buka. What is the disease called? Foolish sapling disease. Which one? Which plant showed that? Paddy. Paddy showed foolish sapling disease. In light reaction, plastoquinone facilitates the transfer of electrons from. Now you have to remember, like you remember the sequence of electron carriers in oxidative phosphorylation, you have to remember the sequence of electron carriers in photophosphorylation also. Electrons are triggered from where? To start with? PS2, photosystem 2. From photosystem 2, who is picking it up? Who is picking it up? Plastoquinone. From plastoquinone, electrons are taken by cytochrome B6F complex. You can make note of this, just a sequence, just a series 1, 2, 3, 4. Photosystem 2, plastoquinone, cytochrome B6F, plastocyanin, photosystem 1, ferredoxin, NADP reductase, and finally NADP. Alright. Photosystem 2, plastoquinone, B6F, cytochrome B6F complex, plastocyanin, PS1, ferredoxin, FNR, ferredoxin, NADP reductase, and then NADP. 2, plastoquinone, cytochrome B6F, plastocyanin, photosystem 1, ferredoxin, FNR, ferredoxin, NADP reductase and NADP. That sequence you should remember. Electron flow. Which of the following is not an inhibitory substance governing seed dormancy? Gibberellic acid, ascorbic acid, phenolic acid, para-ascorbic acid, all of these can inhibit seed germination. Name the enzyme that facilitates the opening of the DNA helix during transcription. DNA? Look at the question again. Name the enzyme that facilitates the opening of the DNA helix during transcription. RNA polymerase only. It has the dual role. It opens up as well as polymerizes. There is no DNA helix involvement during transcription. You get excited by looking at the 80% of the question and don't look at the last word during transcription. This is a good question. RNA polymerase. Otherwise, it is DNA helicase. It opens up. But for transcription, it is RNA polymerase itself which will do the same, which will do the task of helicase also. Dual role. Unwinding and synthesis of RNA strand. Which of the following would help in prevention of diuresis? Diuresis is excessive urination. That means more water is being excreted. What will help in the prevention of that? What will help in prevention of diuresis? Which one? More water reabsorption due to undersecretion of ADH. If there is undersecretion of ADH, will there be more water reabsorption? There will be less water reabsorption. It will promote diuresis. Reabsorption of sodium and water from renal tubules due to aldosterone. Is it correct? It is correct. Aldosterone will stimulate reabsorption of sodium. Water follows. So that will reduce the diuresis. It will bring down diuresis or it will concentrate the urine. Atrial natriuretic factor causes vasoconstriction. Atrial natriuretic factor increases urine excretion, increases urine volume, and its action also is not vasoconstriction. Atrial natriuretic factor will lead to sodium excretion, reducing the volume of blood and thereby bringing down blood pressure. Decrease in the secretion of renin by JG cells. JG cells are juxtaglomerular cells. If renin secretion is decreased, what will happen? If renin secretion is decreased, sodium reabsorption will be affected. Because renin is important for, if renin secretion is decreased, not sodium reabsorption, vasoconstriction is altered. So renin is not regulating urine volume. Renin is regulating blood pressure. Statement 4 is not related to diuresis. So the right choice is reabsorption of sodium and water. In relation to gross primary productivity and net primary productivity of an ecosystem, which one of the following statements is correct? Gross is more or net is more? Gross. Now look at the options. Gross primary productivity is always less than net primary productivity. False. Gross primary productivity is always more than net primary productivity. True. Both are one and the same. No. No relationship exists. There is a relationship. It's a straightforward, easy question. Matching. What do you want to catch hold of? Okay, leading cells, androgen, D1. Done. Option 1 and 2 ruled out. You have eliminated 50% of the options. Then, which one do you want to catch out of again? HCG, placenta. A2, option 4. Placenta, human chorionic gonadotropin, zona pellucida, layer of the ovum, bulbourethral glands, lubrication of the penis, Leydig cells, androgen. Strobili or cones are found in Salvinia, teres, marchantia, equisetum. Equisetum. Pteridophytes have cones. Strobili are compact cones, cone-like structures with sporangia on it. Okay, this is an important question. Good question. You look at this. Some dividing cells exit the cell cycle and enter a vegetative inactive stage. This is called the quiescent stage. This occurs at the end of M phase, G1 phase, S phase, G2 phase. G1 phase. Now at the end of G1 phase, if the machinery is not adequate to cross the G1S checkpoint, if the G1S checkpoint fails, the cell goes into G0 phase, which is the quiescent phase or the inactive phase. Flippers of penguins and dolphins are examples of convergent evolution. Both of them don't have common structures, but the function of both is the same. What kind of organs are they? Flippers of penguins and dolphins? They are analogous organs. Industrial melanism is an example of what? What? Natural selection. Natural selection. If the distance between two consecutive base pairs is 0.34 nanometer and the total number of base pairs in a typical mammalian cell is 6.6 into 10 power 9 base pairs, then the length of the DNA is approximately. You should make a commonsensical logical calculation here. The length of one base pair is 0.34 nanometers and you have 6 into 10 power 6.6 into 10 power 9 base pairs. So total length is the number of base pairs into the distance between adjacent base pairs. So it is 0.34 into 10 to the power minus 9 into 6.6 into 10 to the power 9. 10 to the power 9 and 10 to the power minus 9 cancel each other. So 0.34 into 6.6 meters. Give me a sensible, logical way of calculating this now. You want to write down 0.34 into 6.6 and then start doing the multiplication? Or can you give me a sensible calculation which can do it in your mind without writing down anything on paper? Come on. What is 0.34? If you want to make it into a fraction, 0.34 is 0.34 is 1/3. 1/3 is 0.34. So 1/3 of 6.6 is, instead of 0.34 into 6.6, if you make it 1/3 into 6.6, so it becomes 6.6 divided by 3, 2.2. That's all. No? So fraction to decimal conversions, all of that should be spontaneous. 0.34 is 1/3. Approximately 0.333 recurring is 1/3. 0.34 you can consider for an estimated calculation. 2/3 is 0.67. 0.66, 0.67, that is 2/3. 0.25 is 1/4. Like that. Okay. It makes it easy for you to calculate. 2.2 meters. The QRS complex in a standard ECG represents. Don't look at the options. What all inputs do you get by QRS complex? What all does it speak of? Depolarization of ventricles, systole of ventricles. During QRS complex, what is the phase of cardiac cycle in which the atria will be? Atria will be in diastole. So QRS complex corresponds to ventricular systole and atrial diastole. Depolarization of ventricles, whereas it would be repolarization of atria. After depolarization, atria is going into repolarization. Alright. So depolarization of ventricles. Again matching. This question paper has become a matching center. At least 10 questions are there, no, in this matching question. Some 8 to 10 matching questions are there in this paper. So each matching question is like 4 questions in one question. So if there are more matching questions, possibility of time wastage is high. If there are more statements with which of the following options are correct, 1, 2, 3 is correct, 2, 3 is correct, 1, 4 is correct, those kind of questions also will waste time. So you just have to look at the question. Are they asking you to identify correct statements or wrong statements? Statements, in front of that you put a tick mark for correct statement, into mark for wrong statement. Then look at the options. Don't look at the options and try to match the statements. Okay, which one do you want to catch hold of now, of the 4? Neutrophils. Neutrophils do phagocytosis. That is common. C2. 50-50. Then which one else? Lymphocytes. Immune response. D1. Over, no? Option 1. A3, eosinophils release histamine destructive enzymes. B4, basophils release granules containing histamine. Both are correct. Which of the following statements is correct? Adenine pairs with thymine are 3 options. 4th option is adenine does not pair with thymine. Okay. The difference is 2 hydrogen bonds, 1 hydrogen bond, 3 hydrogen bonds. Adenine binds with thymine with how many bonds? How many hydrogen bonds? 2 hydrogen bonds. That's all. Sequence that controls the copy number of the link-free DNA in the vector is termed ORI site. ORI is origin of replication site, origin site. The copy number is associated with the ORI site. Identify the basic amino acid from the following. AG is basic. Acidic. Aspartic acid and glutamic acid. Aspartic acid and glutamic acid are the only 2 acidic amino acids. HAL, histidine, arginine, lysine is basic. What is HAL? There is a defense manufacturing company, HAL, aerospace manufacturing. It's in Bangalore. Hindustan Aeronautics Limited, HAL. HAL is basic. Histidine, arginine, lysine. All the others are neutral. Aspartic acid and glutamic acid are acidic. HAL, histidine, arginine, lysine are basic. Rest all are neutral. More likely questions come based on the chemical property of amino acids. There is a likelihood of that because amino acids are amphoteric, bitter ionic in nature. They have both acidic and basic properties. Amphoteric molecules. They have both positive and negative charge. Bitter ionic proper molecule. Again match the following. Which one do you want to catch hold of? Catch the easier one. Pancreas, diabetes mellitus. B2, only one option, done, over, fix. Option 3. Pancreas, diabetes mellitus. Pituitary, diabetes insipidus. B, thyroid gland, 1, Graves' disease. Adrenal gland, 4, Addison's disease. You have to tell me the associated hormone also. Pituitary gland, diabetes insipidus, hormone, ADH, antidiuretic hormone. Thyroid gland, Graves' disease, hormone, thyroxine. Graves' disease is what? It is autoimmune. It is not a deficiency disorder or a hypersecretion disorder. It is an autoimmune disorder. Adrenal gland, Addison's disease. Addison's disease is associated with which hormone? Cortisol and aldosterone. Cortisol and aldosterone. Cushing syndrome is? Cushing's is? Cortisol, glucocorticoids. Okay. That's all. Select the correct statement. Glucocorticoids stimulate gluconeogenesis. True or false? Glucocorticoid stimulates gluconeogenesis. True or false? It is true. See, the reason why glucocorticoids, which is a stress hormone, cortisol, glucocorticoid, stress hormone, causes obesity is because of this. It will not respond to glucose being present in the body. Glucocorticoids will add more glucose to the body by converting other proteins and lipids into glucose, trying to make glucose available for utilization. So increased glucose level in the blood constantly, leading to excessive accumulation of glucose as fat by the adipose tissue. Naturally, no, stress is a signal that survival is threatened. Body does not understand psychological stress or ecological stress. The threat is threat. Psychological stress or an ecological factor threatening survival induces the same kind of response. But when there is an emergency, there is other adrenal medulla also stimulated for the release of adrenaline and noradrenaline. But these glucocorticoids are released in response to stress. Threatening survival, prepare the body for better survival. You need energy for fighting bad times. So use all opportunities to make energy available as storage. What is the storage energy that you keep? Lipids, fats in adipose tissue. So reduce, increase glucose availability by converting proteins into glucose, fats into glucose, gluconeogenesis. Push it into adipose tissue for storage. Increases efficiency of storage of energy. Glucocorticoids increases availability of glucose, increases glucose conversion into fat, increases storage of glucose or energy in the form of fat. All of that together contributes to obesity. Stress, obesity. You know, people eat a lot when they are in stress. Stress eating. Stress eating. You also do that when you are stressed out. Not feeling good. What do I have? Chocolate, snacks. Do you notice that when you are stressed, what kind of food do you eat? When you are stressed, do you eat cucumber, tomato, carrot, apple? What do you eat? Chips, chocolate, samosa, fried foods. Why? What you eat at different times of the day or in response to different kind of stimulus is also evolutionary. High energy foods are the craving. With a lot of sugars, crave. The body will crave for that. It will get satisfied with that. Stress is relieved when you eat fatty foods, when you eat high energy glucose based foods. On one side, your body is activating the storage mechanism. On the other side, it is telling you to eat. Push. So what do? Because you have a mind and you have the ability to control your thought and action, whenever you feel anxious, whenever you feel stressed, drink a lot of water. When you drink water, it will settle down any hunger pangs. If you are feeling hungry, hunger pangs will settle down. Many times, lack of water, dehydration, subclinical dehydration is understood by the hypothalamus as hunger. Hypothalamus will be stimulating thirst, but our brain interprets thirst as hunger many times. So drinking water will reduce your craving for eating something. So all of that is good when we are not in that situation, but when we are in that situation, we are driven by our instinct. Instinct will not get satisfied until we eat chips. And plain water will not go in. What do you need along with chips? What do you drink when you are stressed out along with your chips? Sprite, Thumbs Up, Pepsi, Coca-Cola. If I tell you to drink tender coconut water when you are stressed, will you do it? That will actually help. You understand the mechanism? Glucocorticoids. Which one of the following is the most abundant protein in animals? Collagen. Which of the following four are not proteins? Hemoglobin, collagen, lectin, insulin. Which of the following are not proteins? Lectin, no? We have discussed lectin in a previous paper. Lectins are carbohydrate binding proteins, helps in the transportation of carbohydrates across the cell membrane. Lectins are carbohydrate binding proteins, they are also protein. Okay. Experimental verification of the chromosomal theory of inheritance was done by, who did that? Murugan. Mr. Murugan did it. Again, match the following. Matching. Which one do you want to catch hold of? Floating ribs, acromion, scapula, glenoid cavity. Come on, tell me something. Floating ribs, okay. Do not connect with the sternum. A4. Done, over. Option 4. That's all, no? Done. See, this approach saves a lot of time. Otherwise, you will do matching, then look for options. Just one. A4, acromion is of the clavicle. Acromion process of the clavicle. Scapula. Acromion is of clavicle or scapula? Acromion process is of the scapula, to which clavicle articulates. Clavicle articulates with the acromion process. That is still okay. Scapula located between the second and seventh ribs. Dorsally. Glenoid cavity. Head of the humerus. Again, see, head of the humerus does not have glenoid cavity. Glenoid cavity is a part of scapula again. Head of the humerus articulates into the glenoid cavity. So in that sense, both acromion and glenoid cavity are matching with its articular surfaces. Number of substrate level phosphorylations in one turn of the citric acid cycle is 1. In one turn of the citric acid cycle is 1. What is the reaction? Can you tell me? Succinyl CoA to succinate. Enzyme that mediates this. succinyl-CoA synthase, or it is also called thiokinase. Thiokinase. What is produced? GTP is produced. GTP is produced. Alright. If we talk about it as per molecule of glucose, 2 substrate level phosphorylation. Alright. For 1 molecule of glucose, how many substrate level phosphorylation happens in its entire breakdown? The number of ATP, the gross number of ATP slash GTP produced by substrate level phosphorylation per molecule of glucose. What is the gross number of ATP slash GTP produced per molecule of glucose during its hydrolysis, during its breakdown? 2, 4, 6, 8 options. Why 6? 4 in glycolysis and 2 in Krebs cycle, but 2 gets used. If the question was net ATP slash GTP, then it would be 4.